Hello, let's discuss on chapter 11, that is biotechnology. What is biotechnology? Biotechnology is the applications of scientific and engineering principles to the processing of materials by biological agents to provide goods and services. We have discussed about Bt cotton. What is Bt cotton? Bt stands for Bacillus thuringiensis, and this Bacillus thuringiensis is a bacteria. Now, we know that Bt cotton is a transgenic plant, and this cotton contain the genes for this Bacillus thuringiensis. Now this Bacillus thuringiensis provides resistance to insect and bollworm. So this cotton contain the genes for this Bacillus thuringiensis, and the insertion of the genes of this Bt, that is Bacillus thuringiensis, into the gene of this cotton is being achieved by biotechnology. So this insertion is being done by scientific and engineering principles, and once this is the gene of this bacteria is being introduced into the cotton become resistant to insect and bollworms. So this biological agent, that is Bacillus thuringiensis, is a biological agent which when introduced into the cotton, it provides services, that is resistance to this bollworm and insect. So this is all about the biotechnology. Now the DNA that contain foreign DNA in them is called as recombinant DNA. That means recombinant DNA is a DNA that contain foreign DNA with them. So let's discuss on tools for recombinant DNA technology. There are various tools that are required while producing this recombinant DNA technology. Now, recombinant DNA, the first one is enzyme, the second is cloning vector, third is foreign DNA, and the last one is competent host. So these are the tools that are required for developing recombinant DNA. Now the first one, let's discuss on enzyme. What is an enzyme? An enzyme is a biological catalyst that increases the rate of chemical reaction. Now various enzymes are involved in this recombinant DNA technology. The first one is lysine enzymes. These lysine enzymes are those enzymes which help to open the cell or help in dissolving the cell wall of plants or bacteria. So the example of this lysine enzyme is lysozyme. This lysozyme help in dissolving the bacterial cells. Now the next form of enzyme is the cleaving enzyme. These cleaving enzymes help to cleave or cut the DNA molecules into several fragments. And these cleaving enzymes are of following types. The first type is exonucleases. These exonucleases are cleaving enzymes that remove the nucleotide from either 5' or 3' end. That means this is 5', this is 3', 5', 3'. So from either of this end, exonucleases remove the nucleotide. The second is the endonuclease. Endonuclease are cleaving enzymes that cut the DNA from within the DNA at any point, but not at the end. That means it can cut the DNA from here, here, but not from the end. That means it used to cut the DNA from the middle regions of the DNA segments. So these endonucleases cut the DNA from any point within the DNA but not at the end. And also they cut or cleave only one DNA strand. That means they will cut either this one or this one, not both the strand of the DNA. The third type of cleaving enzyme is restriction endonucleases, also called as RES. These restriction endonucleases are those enzymes that cleave both strands of the DNA. Now these are the two strands. that is previously it is joined. Now once restriction endonuclease come into contact with the strands, they will cut the DNA. They will cut both strain of the DNA, and the cutted strain of the DNA become the recognition sites, and these recognition sites contain a palindromic sequences. Now when we say palindromic sequences, these palindromic sequences are those sequences which read same from it when it is read from right to left or from left to right. So if we look at the nitrogenous sequence here C T T A A, so when we read from this side to this side, that means from right to left, C T T A A. So this is the nitrogenous sequence. Now if we read from left to right, C T T A A. So these sequences, when it read from right to left and from left to right, it read the same sequences, same nitrogenous base. So these type of sequences are called as palindromic sequences, and the cut sequence of these DNA strands, they become free and they overhang. So this overhanging free single strand of the DNA that arise when the restriction endonucleases cut the strand of the DNA. This strand, free single stranded strand of the DNA, is called as the sticky end. Now the next one will be synthesizing enzymes. These are those enzymes that help to synthesize the DNA. Now the first one is DNA polymerase. This is an enzyme that synthesizes complementary strand of the DNA on the template strand of the DNA. The second is reverse transcriptase. These are those enzymes that help to synthesize complementary strand of the DNA on the RNA template. The third is ligases. These are those enzymes that help in joining or sealing the DNA fragments. So these are various type of enzymes. Now the second is cloning vector. When we say cloning vector, what is cloning vector? Cloning vectors are those vectors or the DNA segment or the DNA carriers that carries the foreign DNA or gene into suitable host for cloning. Once the foreign DNA is inserted into the vector DNA, the hybrid DNA, that is, hybrid means here DNA vector having its DNA together with the foreign DNA. That means two DNA are present in this vector. So this is called as the hybrid DNA. So the hybrid DNA is called as the recombinant DNA or the recombinant vector. So this recombinant DNA can be introduced into the host cell, such as bacterial cells, so as to produce multiple copies of this recombinant DNA. Now there are various characteristics for this cloning vector. The first one is origin of replication. Origin of replication is the region on the DNA where the replication will start. So if these are the strands of the DNA, and here if replication will start from these points, and this point is called as the origin of replication. Now this origin of replication become the binding sites for the initiator protein to start the process of replication. So for a suitable host for a suitable vector to act as cloning vector, it should have at least one origin of replication so that it can multiply easily inside the host cell. Now the next one is second characters is the selectable markers. These selectable markers, the vector to be used for cloning should have a selectable marker to help in detection of transform host cell. So transform here refers to those cells that have recombinant DNA in them. It is referred to as the transform cell. Now the second is uh, identification and elimination of non-transform. Non-transform means those cells that do not contain recombinant DNA. Third is separating and growth of the transformed cell. So usually the genes that provide resistance to antibiotics such as ampicillin, tetracycline, etc. are used as selectable marker. And bacteria like E. coli do not have antibiotic resistant gene in them, so they do not grow in the medium that contain antibiotics, whereas the transformed cell with antibiotic resistant gene will survive and multiply in the nutrient medium. Now the next one will be characters. The next characters will be cloning sites. Cloning sites are the region where restriction endonuclease will recognize. So therefore a vector to be used for cloning should have at least one unique cloning sites for commonly used restriction enzyme. The next characters is small size. A cloning vector should be small in size because large size molecules usually tend to break during isolation and purification. Therefore, an ideal vectors should be less than 10 kb. Kb stand for kilobase. So an ideal vector should be small in size, having less than 10 kb in size.